Okay, so did you notice that in the video how everything was kind of fluid, how everything was able to move around, and how nothing is static? If you think about it, that really works to our advantage. Having a membrane that is rigid and that is not flexible tends to break easy, i.e. a balloon. Think about what happens when a balloon bumps into something that's pretty sharp or something that's pretty rough and uncharacteristic of a flexible membrane. So if you have any questions about the assembly of your membrane, of course, I'm kind of walking around and just kind of looking at how you created everything and how you set it up and making sure that you've got the correct things that are labeled. So why don't we just take a 2.5 and let's talk about what the arrangement of stuff or the things that you had to look at inside your membrane. So I'm gonna go to another slide. Give me just a second. Let me do some cleaning here. I'm going to make the background dark because it makes it, I think, a little bit easier for you guys to see. Okay, so the first little thing that you guys had to draw, of course, was, let's see. Okay, was this little thing here. And so let's do let's just talk about some introductions here. This is going to be your phosphate head, and this is not a compound that you have not heard of or an anion that you've not heard of. This is, of course, the same phosphate groups that are going to be associated with ATP. Remember, in assembling that bond, that last bond, and this is where the energy from the ATP comes from. Okay, now this right here, these are called the fatty acid chains. And if you remember back from the lipid activity, so I'm going to write this down if you need a refresher, look back over the lipid activity, and we saw that the fatty acid chain, like some of the examples were butyric acid, linoleic acid, oleic acid, lauric acid, palmitic. So I'm hoping that this maybe generates some of that idea. And you guys constructed the triglycerides. So we are just looking at a phospholipid layer here. Okay, so the membrane is made up of a phospholipid bilayer. So let's say phospholipid bilayer. Okay, so it means there's two of them, and they're these phospholipids, the phosphate head and this lipid fatty acid chain, or tails. If you remember too, the phosphates are polar, and the fatty acid chains are nonpolar. Remember, these guys don't like water, and the polar guys do like to have water. And this is what's holding the shape and the integrity of our cell. All right, so the next thing that you guys had to do were some coloring and something. I'm going to try to do this. Please don't laugh. So you had a marker protein. So is that about right? You had to do like a little marker protein. And then, okay, so there's your marker protein. And then we had to do like a receptor. Like a receptor protein. And you had to color that one yellow. And then the other one that you had to do was looked like the symbol for the X game. Okay, so let's kind of. Okay, so that's not quite what it was, but close enough. And you had to embed these things inside the membrane. So these guys are embedded inside the membrane. Okay, now let's take a minute and let's revisit this idea of embedding. In the electron transport chain, you guys saw these channels. And these channels were pumping the hydrogen ions out into the intermembrane space. Well, that's what these guys right here are doing. They are pumping, or are going to be doors that are opening up to the outside of the cell. They're going to serve as channels that are going to allow things to move into it. Now, for the next little lecture that we have, we're going to be looking at some types of transport. We're going to look at passive transport. We're going to look at active transport and some other things associated with the membrane. So see you in a bit.